I always tell students that next time, I always tell myself next semester I will just ditch everything else and we'll just do the entire semester of viruses. When I run out of viruses, then we're probably going to talk about something else. That never happens. But at least I have something that I like for dessert. Viruses. What should I start with? Well, first of all, they are acellular. Remember, we talked about it. How many viruses are there? It's really hard to estimate because remember we had a conversation about species. How we define species in metazoa, in animals. How we define species in animals. If the offspring is fertile or not. You take two dogs of different breeds, it's the same species because the offspring is going to be fertile. You take the horse and the donkey, you get mule in the end. Mules are infertile because donkeys and horses are different species. Does that make sense? With viruses, as with bacteria, there is no sexual reproduction. So definition of species kind of hangs in the air. For this is not going to be an exam. I'm announcing it loudly. Just introduce the concept for you. You may run across it. It's called Operational Taxonomical Unit, OTU. Operational Taxonomical Unit represents all organisms that have 97% or more genetic similarity. That's it. Entirely based on the genetic sequence. Same goes for viruses. We define viral species based on the genetic similarities. So how many species are there? We don't know. Stephen Morse suggested there's about 1 million. I don't know what he was basing his estimation on. The study by Ian Lipkin and co-authors found that a single animal of the fruit bat, and bats are known to carry a huge number of different viruses. Single species of fruit bat carries 58 different viruses. And if we will try to extrapolate it to a number of mammals and then all species in the world with all adjustments, the number of species can be as many as 100 million different viruses. Amount of viruses in the world is gigantic. In the ocean, there is 10 in the power of 31st virus particles. How big is the number? This is very big. I don't know the word for the number. It's comparable to the number of stars in the universe, for instance, known stars. Surprisingly, if you will try to isolate all the virus particles and put them together like in a bowl, all together, all viruses in the Earth, of course it's estimation, will occupy the volume roughly equal to the volume of 2 MetLife stadiums. It's not a lot of volume, but a lot of particles. And another interesting thing, most of the viruses don't cause a disease, whether it's a human disease or plant or insect. In fact, now we know roughly, again, the last data that I saw were for 2011, 219 virus species that cause human disease, out of thousands and thousands of viruses that can infect humans. For example, there is a virus called anellovirus. Not for the exam, no worries. Anellovirus. It infects, not practically, it infects absolutely every person on earth. You can test any blood bank and you will find anelloviruses in every sample. They replicate in humans, we shed them, they cause no symptoms at all, no clinical picture at all. They just use us for the replication, right? Now, viruses not only have no cellular structure, they are obligate intracellular parasites. And if you would look at viruses, what they can and can do, they cannot do two things. They cannot perform the protein synthesis by their own because they have no ribosomes. That's the fundamental feature of viruses. Even bacteria such as rickettsia or chlamydia, the obligate intracellular bacteria, they do have ribosomes. Viruses do not. And they have no metabolism of their own. So they cannot derive energy independently. They have to use cellular energy sources. Does that make sense? Now, viruses can be of different size. Smallest ones, perhaps the smallest virus is hepatitis D. I don't have it. I have here's polio and explain why polio. Hepatitis D, which is going to be roughly at the same ballpark as polio is, with the size of the particles ranging from 20 to 50 nanometers, to the virus like Pandora virus. It's not in this little excerpt, but Pandora virus, to give you a perspective, its size and the diameter is about 700 nanometers. The size of mycoplasma is 200. The virus is almost 4 times bigger than the cell of a bacteria. This virus infects amoebae. And for a long time, it wasn't discovered for one simple reason. The reason was rather stupid. It was observed for years, and it was discovered in 2004, 2005. No reasons? No? Absolutely, they just thought, oh, it can't be a virus possible. It's just, we can see it in light microscope, for God's sakes. Okay? And then they discovered it's gigantic, it's unbelievable. I have, in some other sources I have a picture of that virus using the light microscope. It's like a bullet, a giant 700 nanometer bullet. You can see it without any sophisticated equipment. Now, why I put polio as the small virus? Polio is the smallest important virus. I mean, important. Hepatitis D is, yes, it is important, but it's not very frequent. Polio is the major health concern. Okay. That's why it is often used as the example of small virus. So generally speaking, you cannot see viruses in a light microscope, generally speaking, but there are examples of the ones you can, like Pandora virus. Among the viruses of eukaryotes, more of, not just eukaryotes, viruses of mammals, smallpox, and other pox viruses can be seen in the light microscope. They're pretty large. The size can reach up to 300 nanometers. You can see them, of course, at the very edge of resolution, very edge of microscope capability, but nevertheless. Am I clear? So understand the limits of the visibility. Like that statement, generally you cannot see viruses in the light microscope, but some of them you can. Am I clear? Am I? Good. Structurally, they can be divided in two types, naked and enveloped. Naked viruses, like this adenovirus, consist of the genome and the capsid, which is the protein. Genome is nucleic acid, capsid is the protein. Genome and capsid together are called nucleocapsid. Enveloped viruses, in addition to the nucleic acid and the capsid, have an extra layer of, I wouldn't say protection, extra layer of covering, which is called envelope. Envelope consists of the virus proteins and also phospholipids. You may have noticed that I didn't say virus phospholipids. Where do viruses replicate? Intracellularly. How do they get from cell to cell? I'm asking simple questions. You don't have to know anything. You just have to think. How do they get from cell to cell?
When they successfully replicated in one cell and it bursts, they kind of just float out to the next one.
They get out of the cell and float out to the next one. Cells, what are they surrounded with? Membrane. What does membrane consist of? Phospholipid. That's where viruses, enveloped viruses, take phospholipids. They bud off the cell and take a pinch of a membrane with them. Is that clear? Do I make sense? Awesome. How they were discovered. The first virus that was discovered is tobacco mosaic virus. Surprisingly, virus of tobacco. It's very easy to study and it's very cheap, well, inexpensive to study, because all you need is the bunch of tobacco plants which you can infect. You can see the spots on the infected tobacco leaves. You can compare the phenotype of the spots, you know, like are they bigger, are they smaller, sort of like colonies. Does that make sense? So Russian scientist Dmitry Ivanovsky was curious whether the tobacco mosaic infection was caused by a bacteria or not. So he took an infected plant, mashed it, and then filtered that mash through the special filter that retained bacteria. Pretty much 0.2 micrometer filter. Does that make sense? And then he took whatever went through the filtrate that is bacteria free, and he took this filtrate and used it to experimentally inoculate uninfected tobacco plants. And ta-da, these tobacco plants got infected. So something that was filterable through antibacterial filter, was able to cause tobacco mosaic disease. So he called it a filterable agent. Does that make sense? Because virus could get through the bacteria retaining filter, they were initially called filterable agents. The first animal virus that was discovered was foot and mouth disease virus. It's the significant pathogen of cattle in England. Sometimes I just learned it by the personal conversation with one of my former bosses. She was from England, and she said they were in the tour, like they were on the boat on the river, and they wanted to, how you call it, get to the shore, you know, and so they kind of were approaching the deck, the marina, and people in the marina said, no, you cannot. This is the area that where we have foot and mouth disease epidemic now. You must stay on the boat, go beyond that area, and then you can get in the dry land. Not because people get infected, but because they can transmit it. So, but there's an outbreak foot and mouth disease, in England at least, they call the entire herd, just every animal. We talk about millions of animals often. Okay. So how can we classify viruses? First of all, we can classify them by being naked or enveloped, right? That's the common. Have you heard about Can we say group of respiratory viruses? Can we? I just said it. Right? We can, of course we can. Of course we can say. We can say viruses in enteroviruses, viruses that cause enteric disease, respiratory viruses, blood-borne viruses. Okay? So we can classify them by the symptoms, by the transmission, like arboviruses. Arbo stands for arthropod-borne. Viruses that are transmitted by arthropods, ticks, and mosquitoes. But it turns out that in every group, viruses can have huge differences between them. That makes sense? They can be really, really different. So currently the classification is based on the genome. Little digression. Hopefully you'll find it mildly amusing. Next time, when you hear about science conspiracy, think about this. Somebody finally suggested, here's the great classification based on the genome. Immediately, there is a scientist that said, no, I have better. Scientists cannot agree on anything, even that anything benefits them. Take 10 scientists, tell them to order lunch. They will starve to death before they will all, agree on what they have to order. So they cannot possibly conspire. Viruses now are classified based on the structure of the genome. And without going too much into the different replication strategies, viruses can have both DNA or RNA, not N, but or RNA as the genome. DNA genome can be double-stranded, like we have. So typical double-stranded DNA. Herpes viruses have that genome. Some viruses have one strand of DNA, like parvo. And viruses can also have RNA carrying genetic information. Double-stranded RNA, like in rotaviruses. Positive or negative single-stranded RNA. Positive is the Zika virus, negative is the Ebola virus. What do you think positive and negative mean? We didn't talk about it. This is the tough question. This is not a simple question. I'll give you a little hint. I will write down the central dogma of molecular biology. What is the positive RNA? What do you think? Any thoughts? Come on. You just nailed it. That's positive RNA. If RNA molecule is used for protein synthesis, it is by definition positive. What is negative RNA then? I'm going to ask you, so here's the little question. Technically, if you have just hypothetical RNA, can you translate it? Can you make protein using that RNA as the matrix? Yes. So technically, any RNA can be imagined as positive. But in viruses, I'm trying not to give you a clue. So positive RNA is the one that virus uses for protein synthesis. What would be negative then? Very basic feature of nucleic acid, either DNA or RNA. Two strands of what? Two strands are complementary. Negative strand is complementary to positive strand. you can use positive strand for protein synthesis right away. If virus has negative strand RNA, it has to make a complementary copy of it, which will automatically be positive, and then use it for translation. It's like, you know, like when in old times when you used film for pictures. Did anyone use film for pictures? It's making photos. Huh? It's a negative, yes. So you pull out the film and it's so funny to look at it. Because people's faces like dark, and people like light clothing is dark, okay? And then dark clothing is white, and it's all reversed, right? So that's negative. And then in order to make a picture, you need to, so it's a next step, it's an extra step. So negative RNA is copied, complementary copied into the positive one. And then this can be used for translation. Does that make sense? Do you understand the idea? I'm going to ask positive and negative RNA, I promise. Do you understand? Okay. Structures of the virus capsids. They're different. I'm going to talk about structures and wrap it up. The most common structure is icosahedral. Sounds like a tongue breaker. In fact, if you want to imagine something close to icosahedral shape, take a soccer ball. That's pretty much what it is, what it looks like. Very similar. Other shapes, capsid can be helical. Okay. Like tobacco mosaic virus. Some viruses, whether they are mammalian or bacteria, viruses of bacteria, can have complex capsids, like this pox virus, which has extremely complex capsid, or bacteriophage, which looks like a tiny alien spaceship, which capsid has icosahedral head, the tail fibers, the tail pins. Okay, this is all virus. Okay, so notice complex shaped capsids, helical capsids, and icosahedral capsids. One thing that I want to tell you before I let you go, it's not on the slide, but this is the piece of information that I absolutely want you to know. There's nothing complex about it. It's a very bold statement. Viruses can infect everything. Which means we now know about viruses of amoeba. I'm just, whatever pops in my mind. Amoeba, algae, plants, humans, pretty much all mammals, fish, reptiles, amphibians, clams, mollusks, different types of mollusks, insects, of course, tons of them, bacteria, archaea. We know about viruses that infect human parasites. There's a human parasite called Leishmania, and there is a Leishmania virus that infects Leishmania parasite and makes disease worse. So if there is an organism for which we don't know any viruses, my presumption, we didn't look well enough. They are there. Okay? We'll see you tomorrow.